Hello everyone. This presentation will go over caffeine synthesis utilizing a cell-free protein synthesis system completed by Group 1.
Hello, my name is Miguel Antonio Hesse, and I will be explaining the background portion of this presentation. So, essentially, natural products are just a big category of small molecules that are very important to the pharmaceutical industry. However, many companies decommission their small molecule discovery programs in favor of combinatorial chemistry. Um, and the reason for that is that combinatorial chemistry seemed like a faster, more automated alternative. Companies wanted higher productivity and quicker returns as pharmaceutical companies are under constant pressure to find new drugs quickly. So, combinatorial chemistry has appeared to offer a way to speed up discovery and cut costs. So, many companies shifted their focus and shut down their traditional small molecule programs that looked quote unquote inefficient by comparison. However, the shift didn't really pay off as over time researchers realized that combinatorial libraries often produced many compounds with poor drug-like properties. Essentially, they just didn't perform well in real biological systems. So again, like this kind of led to um, an interest in the more traditional uh, chemistry and natural products are kind of being more explored today as more viable alternatives, as shown in this study. Hello, my name is Miguel Antonio Hesse, and I will be further explaining the background. As I said in my last slide, natural products are very large and complex, and that's why a lot of these pharmaceutical companies kind of catered more towards combinatorial chemistry. But the thing with a lot of these natural products is that they contain inherent biological activities, which just works better in these real world biological systems. Examples of this would be erythromycin, daptomycin. A big one today is Taxol, which is used in the treatment of many different cancers, such as lung, breast, prostate, etc., etc. And the thing with these products is that they contain like a very important methylation step that is very crucial in the understanding of methyltransferases in cell-free protein synthesis systems. Cell-free protein synthesis systems have numerous applications in natural products, and there's many studies that have used these cell-free protein synthesis systems, such as chlorogenic acid and the others listed in this slide. However, it has not yet been explored in the synthesis of natural products from plasmid or precursors, which pertains to this study. Hello, my name is Miguel Antonio Hesse, and I will be doing the final slide for the background. So, as I was talking about in my last slide about CFPS systems, the most common system is an E. coli based system. So, essentially, the enzymes of interest, in this case, it would be the TCS1, the T caffeine synthase, um, would be overexpressed in E. coli. Then the CFPS extract is prepared for the culture and then eventually utilized to make the desired product. So eventually that just begs the question of why caffeine? Again, caffeine is a useful and natural product that's created by MT ACEs. And then there's already established methods of caffeine purification and identification using liquid chromatography mass spectrometry. And then this kind of brings me back to the T caffeine synthase enzyme, or TCS1. It's an enzyme that is expressed well in E. coli, and then it contains two different methylation steps in the production of caffeine in T plants. Um, so essentially, all in all of these factors make caffeine an ideal candidate for further exploration and demonstration in CFPS systems for product synthesis.
This is Joaquin, and I'll be presenting on experiment design. So the experiment design for this paper had 6 major components. Some fall under others, but it follows like this. First was the preparation of the plasmid. Then it was cell-free protein synthesis, so CFPS, and its purification. Then it was caffeine synthesis with the purified TCS1 product from that purification. Then it was synthesis and extraction from the CFPS system. Then liquid chromatography mass spectrometry. And finally, HMAT2A protein synthesis and its purification. So with the plasmid, the researchers made a plasmid with the T-caffeine synthase 1, so TCS1 gene, via codon optimization to express caffeine synthase from Camellia sinensis in E. coli. And this was done with integrated DNA technologies, their codon optimization tool. They ordered it as a G block to put it in a different type of plasmid, is PNIC28BSA4. Then they transformed that plasmid into a CH5 alpha E. coli strain, which could then make more of that same plasmid. Then for the CFPS, on the lysate from that E. coli strain, the researchers did CFPS with the Bioneer Exaprogen protein synthesis kit. Protein was synthesized. The protein was purified with nickel nitrilo acetic acid, so nickel NTA beads, and then mass was observed with SDS page and nanodrop spectrophotometry to quantify that concentration. Finally, for the synthesis, an in vitro reaction buffer was done with tris magnesium chloride, S-adenosylmethionine, SAM, and theobromine, and it was mixed with a purified TCS1 protein from earlier. The product was then extracted with chloroform and analyzed with liquid chromatography mass spectrometry, LCMS, to verify the presence and function of the TCS1 protein. In caffeine extraction from the system, they first had to make caffeine in the system, which was done by adding theobromine and SAM with the TCS1 plasmid. Then chloroform was used to purify the caffeine, and they removed the chloroform with pipettes and evaporation, and this left the pure caffeine for subsequent LCMS analysis. And that's how they identified caffeine production was LCMS. And they primarily used ion chromatograms to show species at an expected mass to charge value, which was around 195.1 for the presence of caffeine. And even though there were impurities, the reason they did this was to more closely look at the species of interest that they were able to isolate, um, versus if they used a wider range, it would be more difficult to really look at that. And then finally, they had to produce HMAT2A, which is human methionine adenosine transferase, and they obtained that to be able to make SAM, which was used in the extraction earlier. This is obtained by transforming HMAT2A plasmids into Rosetta E. coli cells. These were then incubated, centrifuged, lysed, and passed through the nickel NTA resin. And the protein was then eluted, verified with SDS page to verify identity. And then it was used to make the SAM with TCS1. This was done to determine the specificity of TCS1 for SAM or for 8-OET, which was S-adenosylmethionine, when SAM is made in vitro as a coupled reaction in CFPS.
Hello everyone, my name is Mariam Lopez and I will be presenting the key results section of this study. To begin, the team successfully expressed the T-caffeine synthase enzyme known as T-caffeine synthase 1 or TCS1, using a cell-free system instead of living cells. The SDS page results show a clear band around 43 kilodaltons, confirming that the enzyme was produced. The yield was approximately half a milligram and remained stable even when the substrates or cofactors were added, showing that these components didn't interfere with expression. When the purified enzyme was combined with the cofactor S-adenosylmethionine or SAM and the substrate theobromine, the researchers detected caffeine using liquid chromatography mass spectrometry, confirming that TCS1 was active in the cell-free reaction. Interestingly, when they substituted theophylline as the substrate, almost no caffeine was produced, which show that T-caffeine synthase 1 is highly selective for theobromine. These results confirm that caffeine biosynthesis can be successfully replicated in vitro, making a significant achievement for synthetic biology. The next key experiment tested whether caffeine could be synthesized directly within the cell-free reaction without ever purifying the enzyme. In this one-pot reaction, the plasmid encoding TCS1, the substrate theobromine, and the cofactor SAM were all added at the start of the cell-free protein synthesis process. After incubation, liquid chromatography, mass spectrometry analysis show a clear caffeine peak at a mass to charge ratio of 195.1, which matched the caffeine standard. The total yield was roughly 10 micromolar, demonstrating that the enzyme remained active even while being synthesized in real time, therefore concluding that the cell-free system could both produce the enzyme and generate the final product simultaneously, something that will normally take multiple steps using traditional methods. The researchers then explored whether T-caffeine synthase 1 could accept a modified cofactor, S-adenosyl-athionine, or ADOT-ET, instead of S-adenosyl-methionine, or SAM. This test was designed to see if the enzyme could create new ethylated caffeine analogs. However, no product was detected when S-adenosyl-ethionine was used, indicating that the T-caffeine synthase 1 is highly specific for S-adenosyl-methionine and lacks cofactor flexibility. In conclusion, this study demonstrates that caffeine can be synthesized completely outside of living cells using a cell-free protein synthesis platform. This is a major shift for translational biosynthesis, where enzymes must be expressed and maintained inside living organisms. The cell-free approach allows scientists to quickly test enzymes like T-caffeine synthase 1 and modify conditions without worrying about cellular toxicity or metabolism. Although T-caffeine synthase 1 was active with S-adenosylmethionine and it couldn't use the ADO-ET analog, showing that it was a strict substrate specificity. Still, this provides a strong fundamental for engineering methyltransferases to accept new cofactors and make novel caffeine-like compounds. Beyond caffeine, these works highlight how cells-free protein synthesis system can be used for rapid prototyping of other natural products pathways. This type of platform could accelerate the discovery and testing of new molecules for pharmaceuticals, flavor compounds of bio-based materials, all while avoiding the limitations of traditional cell-based production systems.
My name is Carmen, and in this section I will discuss the significance and broader impact of this research. This study is groundbreaking because it shows for the first time that caffeine can be made entirely outside a living cell using a cell-free protein synthesis system and the enzyme T-caffeine synthase, or TCS1 for short. This is very important because it means we can now recreate parts of the metabolism without depending on an organism to do the work. Normally, producing natural products in cells is difficult because toxicity, slow growth, or competing metabolic pathways often interfere. The cell-free system bypasses all of that by allowing us to control every reaction component directly. Remarkably, this method combines plasmid DNA, substrates, and cofactors in a single reaction tube to produce the product. In doing so, it shifts CFPS from just making proteins to actually creating small molecules, representing real biosynthetic chemistry. Beyond the technical achievement, this study opens a lot of doors for how we discover and make new molecules. Because the reactions happen outside of cells, we can quickly test enzyme variants or pathways combinations. Basically, prototype new molecules overnight instead of over months. It's also a greener, more sustainable way to do biomanufacturing. We don't need antibiotics, sterile culture rooms, or GMO containment setups, which makes the process both cleaner and simpler. Another advantage is that cell-free systems don't die from toxic compounds. They can handle cofactors like S-adenosylethionine that would normally kill living cells, letting us make analogs and explore new chemistry. And because CFPS is modular, it fits perfectly with automated platforms and AI-driven optimization, paving the way for truly programmable biomanufacturing. It is fascinating how this study changes the whole approach to synthetic biology. Instead of engineering a microbe and waiting weeks to see if it works, CFPS allows you to go directly from plasmid to a functional pathway in a day. Essentially, this study transforms biology into an open chemical system. No cell membranes, no interference from metabolism, just pure biochemical control. That means you can test toxic or non-natural substrates without the risk of killing your host. They even couple two enzymes, TCS1 and human methionine adenosyltransferase, to demonstrate multi-enzyme reactions occurring outside of cells. This is a major step towards creating customizable, cell-free metabolic systems. In summary, this work pushes synthetic biology into a new phase that some call synthetic biochemistry, where the chemistry of life becomes a flexible, programmable toolkit. Although this study is conceptually groundbreaking, it still encounters technical challenges. The caffeine yield was relatively low, about 10 micromolar, mainly because of inefficient extraction and suboptimal reaction conditions. This indicates that further optimization is necessary for broader industrial relevance. Future work could implement continuous flow CFPS systems that constantly supply substrates and remove products, enhancing yield and scalability. Engineering methyltransferases to accept alternative SAM analogs could also increase product diversity and reaction efficiency. The main advantage of this approach is its role as a quick in vitro prototyping platform. Promising molecules can initially be tested in CFPS, then later transferred to engineered cells for mass production. And lastly, because CFPS is safe, cell-free, and inexpensive, it offers an ideal educational and innovation platform for biochemical experimentation.
My name is Sky, and I will be going over the limitations of this study as well as objectives for future studies. So the researchers had a low yield of 10 micromolars of caffeine. So this could be due to the extraction process. They used chloroform extraction coupled with liquid chromatography ultraviolet mass spectrometry. The enzyme that they used was T-caffeine synthase, which has a low substrate specificity. They did not optimize conditions such as pH, temperature, and nutrient concentrations. And then they also used theobromine, whereas previously it had been hinted at that theophylline would be a better substrate. So there is the opportunity for optimizing this pathway by correcting those limitations. So this research was really just a preliminary study in using cell-free protein synthesis systems. So there is a lot of room for future studies. As mentioned previously in the limitations, I will just quickly overview some opportunities for future studies. So one of the things mentioned was to refine extraction by either refining the chloroform extraction that they use or using alternative pathways such as percolation, reflux extraction, or supercritical fluid extraction. Another way is modifying the enzyme to make T caffeine synthesis 1 to use SAM analogs in a coupled reaction or using alternative MTAses such as the two listed here. Another way is to optimize the conditions by either using an alternative substrate and then additionally modifying the pH and temperature to really optimize the production of caffeine in this system.